And e-learning outcomes for this lecture would be understanding the concept of oncogene and tumor suppressor genes, and what are the different types of genetic changes that occur in these two types of genes, and what are the biological processes involved for these two types of genes. And I will give you lots of examples of genetic changes in cancer, either at the chromosomal level or at the DNA level with mutations, either in the context of predisposition in cancer or sporadic cancer, which is also somatic mutations. So just to give you a bit of history of cancer, which is always quite interesting to know, Pierre-Paul Broca, who is a French clinician, and he was interested in pathology, anatomy, and surgery, actually published in 1866 treatises on tumors. And at the time, the concept of genes was not known, but what he found out is that his wife and his family, lots of different generations had breast cancers. And so he understood that there was something which was transmitted over the generations to the offsprings, but then it must have been latent because a daughter who was born years after a mother or grandmother took years and years before to develop a breast cancer. So something was hereditary, something was transmitted to the generations, it was latent, and it was later in the life that cancer occurred. Later on, another scientist, and actually a German biologist, Theodor Henrik Boveri, developed a theory which is actually still in actuality because it's the basis of genetic cancer, but the somatic mutation theory of carcinogenesis. And he actually published that in 1914, and you'll notice that it's just a year before he died. He died quite young. And again, at the time, there was no concept of genes, but he was working on sea urchins, and he understood that there was some cancers, they were happening from one single cell. So cancer, tumors, it's a clonal disease. So it originates from one cell, but had acquired a genetic change, and then later on, this cells acquire more mutation, as you will see later on. But then this, he described this single cell having a makeup of chromosome which was completely scrambled, and it was due to aberrant mitosis. And he also showed that the cells, the causing of cancer was the cells divided uncontrollably, so there was uncontrolled growth. So I actually have a little quiz for you. Do you know how many genes we have in the human genome? And do you know how many cancer genes are known so far? Does anyone of you have an idea? No? So we have approximately 24,000 human genes. And actually, it's way less than we thought at the beginning. Before the genome was sequenced 10 years ago, we thought we had 100,000 genes. And actually, the reason why we have only 25,000 genes is because all of these genes are tightly related. So there's a lot of uncoding sequences, like non-coding RNA, that regulate genes. And one gene doesn't necessarily give rise to one protein, but can give different isoforms. So we are very complex organisms because we also have not only lots of genes, but we have tight regulation of these genes. And we have approximately 1% of our genes which are mutated in cancer. So there was this group in Cambridge, I don't know if you know in Cambridge, the Sanger Sequencing Center. They have they've done a census of all of the cancer genes in the genome. They sequenced thousands and thousands of different types of cancers, and they found that 1% of the 24,000 genes can be found mutated. basically, cancer is a genetic disease, and there is a variety of changes that can take place. So mutations that may occur in a gene that will directly lead to cancer, but also changes may occur in genes associated with the regulation of cell processes. And you'll see some of these cell processes today, but you'll see also in the next following weeks, like cell cycle or apoptosis. And these genetic changes, they can be either heritable, with predisposition to breast cancer, as an example, or they can be somatic. As every cell must make an exact copy of its 6 billion base pair genome, which is the size of the diploid cells, before it divides, and the polymerase that copies the DNA has an intrinsic error rate, then mutations arise at random, and the rate of mutation increases with the number of cells that are dividing. So you understand that cancer is an uncontrolled growth, so the cells keep dividing. There is no signal to say to the cells, or where there's a certain type of mutations, you have to stop and repair the mutation, and then you can really start to divide. Then in cancer, this control is lost, so the cells keep growing, so then eventually they also acquire lots of mutations. So how I've just introduced just before, mutation can be somatic or germline. If it's a somatic mutation, it occurs in the non-germline tissues. So it can be either on your skin cells, or it can be breast cells. And this you don't transmit back to your offspring. So it's a somatic mutation, it's a sporadic cancer. As opposed to that, you can have a germline mutation. So it's a mutation that is present in the eggs or in the sperm, and you will transmit that to your offspring, and that causes cancer to be in families. So it's a family syndrome. And then all of your offspring, there will be 50% chance of your offspring to inherit the mutation. Yeah.
You know, you've given the example for the somatic mutation as breast cancer.
Yeah. So
can breast cancer be a result of germline mutation as well?
Yes. So I'll give you later on the.
Is it only somatic, or in terms of?
So in terms of breast cancer, it's only less than 10% of breast cancer which are familial, hereditary. So 90% of the breast cancer, it's a somatic mutation. So it's two mutations that are acquired in the breast cells.
So if I can just add, so basically, so most breast cancers are just by bad chance, where you get a mutation, something happens, cancer causes an event, and it causes a mutation. Whereas about less than 10%, as Anna said, are down to inheriting faulty genes.
Yeah.
Yeah. So the germline mutation is where you have a faulty gene that you've inherited. Okay.
Thank you.
Thank you. So oncogenes. So oncogenes are actually the mutated version of proto-oncogenes. So proto-oncogenes are genes that we have in a genome, and they are not mutated. And they code for proteins that regulate cell growth, differentiation. And actually, to give you a bit of history, the first proto-oncogenes were discovered in 1910 by Penton-Rose, which actually was working on sarcoma. He was working on chicken sarcoma. And what he found out is that when he extracted the tumor from the chicken, he smashed the tumors and he took off the cells and he kept the supernatant, and he re-injected that in new chicken which were not having any cancers. And eventually this chicken developed cancer. So he was able to show that actually the cancer was not transmitted by the cells, but it was transmitted by viruses and RNA viruses. And so the first discovered proto-oncogenes were coming from genes in the viral particles, and they were called VSARC or VMIC, VJUN or VFOS. And then it's only later on in the mid-70s that actually we found out that these viral genes were also existing in the human genome, and we called them C, so C for cellular, and V was for viral. So we found out that we had the equivalent of VSARC in human, which is called CSARC, and the same for MIC, JUN, and FOS. So we have CMIC, CJUN, and CFOS. And so these proto-oncogenes are unmutated in the normal cells. So oncogenes are this mutated proto-oncogene, and it's actually a gain of function. It's a mutation. It can be a DNA mutation or a translocation or an amplification, and I'll give examples later on. There's like around 14 of these oncogenes which are involved in the majority of cancers, and they stimulate cell division, inhibit differentiation, and they block cell death, or also known as apoptosis. So when they are mutated, they promote cancer. There are three major types of genetic alteration leading to oncogene activation. So as you see, so you have a proto-oncogene with a promoter region, and either you can have a point mutation, so it's like at the DNA level you have a nucleotide that is changed for another nucleotide, for instance A that becomes a G, or a C that becomes a T. Or you can have a translocation, which means that a part of the gene is fused to another gene, and that has for consequence to create a fusion protein. And sometimes this gene is next to another gene who has a different type of promoter. So then a gene that is not supposed to be turned on, all of a sudden, because it's fused to another gene that is constitutively on, then your gene, instead of switch off, becomes switch on. Or you can have also an amplification, so you have several copies of your genes in your genomes. So, to give you some example of oncogene, they are dysregulated alternating cell signaling pathway. So what is cell signaling? It's basically a signal that comes from the outside of the cells, and it could be a growth factor that binds to the receptor, which is a growth factor receptor. And there is a cascade of molecules, of signaling molecules through the cells, who gives a signal to the nucleus for some genes to be turned on or to be turned off. And you can find examples of oncogene all over the way through the cell cycle. So you can find some growth factor receptors which are amplified in breast cancer, and for instance, RBB2 is an example of oncogene that is amplified. Or you can also find EGFR for small, non-small cell lung cancer. So when this receptor is turned on, it gives a signal to some proteins to go to the nucleus and turn on some genes. Some of the oncogenes, for instance, are RAS or RAF, and you can find also some tumor suppressor genes that I will describe later. So tumor suppressor genes, so the normal function is to inhibit the cell proliferation and the survival. So often we give the example of oncogene and tumor suppressor genes with an analogy to a car. So an oncogene is actually the accelerator in the car. So it has acquired a mutation, and then it's on all the time. And the tumor suppressor gene is actually the brake of the car. So the tumor suppressor gene, its normal function is to inhibit cell proliferation and survival. Oncogene, they are here to actually, they are involved in cell proliferation and in differentiation. So an oncogene, I said earlier, is a gain of function because you only need one mutation in one of the alleles. You know, when in your cells you have two copies of your gene, which are called alleles. A tumor suppressor gene, you need to have the two alleles to be mutated. And when it's a loss of function, so an oncogene you have gain of function, and a tumor suppressor gene you have lost the function. You have lost the capacity of inhibit cell proliferation and to protect the cells against mutations, for instance, DNA repair. Some tumor suppressor genes are involved in DNA repair. So here is an example. It's a table. There's lots of different genes, but to show you, in the outcome learning, I told you that you will see in which biological processes these oncogene and tumor suppressor genes are involved. So you can see that some of these tumor suppressor genes are involved in cell division, in controlling genes. They are involved in DNA repair, like I've explained with BRCA1 and BRCA2, which are the breast cancer genes. You can also have tumor suppressor genes which are involved in programmed cell death, which is also called apoptosis. And I think next week you're going to have a lecture on cell cycle and apoptosis. And here, that's quite an interesting table. I put it here for you because it also shows you that some of these tumor suppressor genes can be either involved in inherited cancer, but they can also be involved in non-inherited cancer. So for example, APC, I will give an example of APC, familial adenomatous polyposis. It's the inherited form of cancer when you have a mutation, a germline mutation in APC, but you can also have sporadic mutation in APC who gives colorectal cancer. I wanted to give you as an example one of the major tumor suppressor gene in cancer, and this is called ERBB for retinoblastoma. And actually, to give you also a bit of history, that's the first tumor suppressor gene that has been discovered. And it's been discovered in the late mid-70s. And it's been discovered by Alfred Knudson, who was interested in this type of cancer, which is called retinoblastoma. And it's a tumor of the retinal cells. And there is two forms of this cancer. There's 40% of this cancer, which is germline, is due to a germline mutation in the gene called ERBB1, and there's 60% of the other tumors which appear in a sporadic context. And he has found that some of the patients with inherited retinoblastoma, they have the cancer at an early onset age. And this is one of the characteristics of cancer that are in predisposition in familial disease. As a familial disease, is that the age of onset is earlier than for sporadic. So if, for instance, if you have a retinoblastoma in a familial context, you'll have the tumor when you're a baby or when you're a very young child. But if it's in the context of sporadic, it's going to be later when you're a young adult or an adult. And this is due to the fact, and this is what makes me introduce you the two-hit theory that Knudson described in 1971, is that when you have a sporadic cancer, because you have two alleles, you need to acquire two mutations in your two alleles. And that takes time for your cells to acquire these two mutations. But if you think that you have already inherited one of your copy, and here, for instance, the red copy is already mutated, so you only need one or more mutations to develop cancer. So then that's why in the context of predisposition, you have cancer that happens earlier in your life than when it's sporadic, because you already have inherited from your parents, from one of your parents, a mutated copy of the gene. Another really important tumor suppressor gene, which is found to be the most frequently mutated gene, is P53, and it's called TP53 for tumor protein 53. It's a transcription factor. and it's a protein of 53 kilodaltons, hence its name. So it prevents the cell from completing the cell cycle, and it prevents